Okay, welcome back. Also, people that are connected in online version, welcome back. We will begin our conference shortly. Cik mēs vēl esam? Jāgaida. Yes, so for this part, we will have 4 speakers. One will be here and 3 will be all online. So, and after that, at 1, we will have another lunch break. So, okay, maybe there's more people. And without further ado, yes, I will welcome Sabrina from Uzbekistan. And this. Oh, and what the back also? Yes, okay, yes. So here is the pot. It's bottom floor. Yeah. So yeah.
Good morning, professors and participants. We are from Uzbekistan. We are students of Samarkand Agri Innovations and Research University. My team member Rahmatullah Sabrina and me, Kamjan Fatabek. Our project is about in vitro cultivation of sweet potato. Let's start our project. To develop and implement and in vitro propagation technology for sweet potato, enabling the production of high quality disease-resistant seedling. This method aims to enhance crop productivity and promote sustainable and eco-friendly agricultural practices there to demonstrate the good effect of the release. Nowadays, the world's population is increasing day by day. At the same time, people's need for food is also increasing. From this, we can see that we need to supply the world market with the food we consume based on the level of human demand. We are currently working on the in vitro propagation of sweet potato plants using the conditions available to us.
We know global and climate-related issues. Climate change and drought, rising temperatures, and water scarcity negatively affect agriculture. And then soil degradation in agriculture. And declined soil fertility and salinization make traditional farming challenging. And then food security challenging. With the growing global population, the demand for food is increasing. Sweet potatoes rich in nutrients can be valuable food source.
Yeah, good point.
And solution to these problems. We know sweet potato strong root systems help us prevent soil erosion. Alternative growing methods like hydroponics and aeroponics can also be used. In vitro propagation allows rapid seedlings production, helping to reduce food shortages. Producing high quality and naturally selected seedlings without genetic modification. The picture is growing in vitro laboratory. This plant, sweet potato, is many system culture put on this medium only. Rooting systems, this acclimatization picture. Annual seedlings production. Okay, cycle one, we need 100 plants with cutting and preparing 500 and 1000 seedlings. Cycle two is 8 months, 500 or 1000 seedlings. We preparing just two 2.5 hundred and then 10,000 seedlings. Annual production estimate under optimal condition. This is number. Thank you for that.
Thank you.
Do you have any questions, maybe?
So how many varieties you propagate in this way?
Propagating this protocol.
No.
Varieties.
Varieties.
What
How
variety?
many different sweet potatoes?
Thanks for watching.
Yes,
Only one time.
yes, only one. Perfect.
And okay, you got these seedlings, do you have some yield? Do
Kā
you
es viņu
also
varu
have,
redzēt lielā?
Tubers or,
Tubers.
yeah, but how much?
How much?
Yeah.
In Uzbekistan, maybe now is 500.
5,000 tane.
Yok, 500 daha fazla en. Şey var ki
500 tubers.
Okay, from these, from how many seedlings?
Kaçta kuşattı?
işte kucaktan.
From one tubers, kaçı kuşat? Neşte? 50 diyeceğim. 5,000 to 50,000.
Okay, you get,
100 tubers. I just take out versus the cutter and then 500.
this, okay, that's 5,000 seedlings, okay.
In how long time?
8 months.
Like 8 months?
Yes. Yes.
You-
Preparation.
You show the rotation in every 4 months. You propagate, but where do you put them? Because can you grow them during the winter season? Because if you grow the seed every 4 months propagation, then you must put them somewhere until the planting season. So isn't there just a time frame that you wouldn't grow them? Or the
Yes, only the in vitro laboratory is not very hard.
So you growing more and more and more,
Yes.
and then in the February or January, put all them to acclimatization.
Yes.
Okay.
Because I have a question about the components in this liquid for roots. Only minerals, you have already some other additional substances too in this liquid.
Kızlar suyuculuk takibini var fakat minerallanmıyor, başka nasıl da bulabilir.
For vitamins, no, some guard hormones and vitamins.
If that's it, then thank you again. And the next speaker is from Ukraine, Bulduri and Larissa Semenko. I will stop sharing. And so,
Hello.
yes, hello, we can hear you.
Yes, sorry. I need 2 minutes, please.
Okay.
Now sharing my presentation. Yes, we can see.
Yes, we can see it.
My topic, identification of promotion clones lines of potato, varieties Belarosa and Rodina by a complex of economical valuable transactions. Oh, sorry. In this work, clones line Belarosa and Rodina, we used the statistic methods. The effectiveness of plant micropropagation is also demonstrated. Improvement of potato varieties zones biotechnology. As a result, good selection of productive virus-free lines. We have genetics identical matter, quickly propagation, presentation of unquiet characteristics. Also, we can see two photos in this source. Materials and methods. First, in vitro potato lines need adaptation to ex vitro. We studied stem length, leaf area, the mass of tubers, the number of tubers, the radius of the mass of each to the mass of tubers, as well as the mass of clones. The coefficients of varieties served as a basis for our constellation and basis to the analysis of data. We were able to identify the most homogeneous lengths for future research. In this slide, we can see technology, cultivation of potato in vitro, first selection of the search plant, next allocated of meristem, culture intentional work in a laminar box, also controlled conditions need stated temperature, usually 20-25 degrees. Also photo period 8 sun and 8 black period. Also, 5 plants need surf in period testing. Also, adaptation for plants to ex vitro. We can see a photo. Representation of transportation, planting in the stub state. Also, a potato in a wheel. Also, next slide, planting in the soil. This card map, but in Ukrainian language, sorry, you choose a place, soil preparation, planted sinking. It's very important. Result and decision. Stem left, the area of the first left. Sorry.
Йдіть в ту переговорку, бо там мене не пустили, мене там зайнято було. Будь ласка, я дуже прошу.
Basis on the assessment of biometric indicators across as characteristic for Belarosa variety lines, the following constellation can be drawn. First place, we have line number 11 and 13, exhibit 8 positive indicators out of 8 possible. Also, second place, lines number 7, 8, and 8, we have 7 positive indicators. Third place lines number 7 and 10. We have 6 positive indicators. Based on the assessment of biometric indicators across all characteristics for Belarusa volatility, we have first place lines number 6 and 7. We have 6 positive criteria. Second place lines number 6 and 8 positive. Second place, sorry, 3 and 5 lines. We have 5 positive criteria. The yield, stability, and stability of lines are selection characteristics. In future work, we recommended to change the moisture level and apply the impact. Conclusions. The results of statistic methods are useful. We can identify strong clonus lines. Equitative phenotype is important for the productive for potato lines in the since production. Thank you for your attention. Sorry.
Thank you. Does anyone have any questions?
May I ask a question?
Yes.
Thank you for your interesting presentation. Will you check the virus precondition or the virus components of the main tubers or let's say the settings in metro condition before planting it to the net house or field?
Which virus? Sorry, I repeat your question.
Now you talked about the in vitro condition, in vitro laboratory, and then you grow first in vitro, and then you transfer it to the greenhouses and then to the open field.
Yeah. First in vitro, next step ex vitro. In the end, we have plant area in the field.
My question is related to this in vitro. Do you check also the virus components of seed in vitro condition?
No, any virus we don't have in this process.
Okay.
Any more questions? If not, then yes, thank you.
Thank you.
Okay. So I would like to invite the next speakers from Uzbekistan and Korea, Udaya Munira and Yoon Yeo Yoon. So let's welcome them.
Okay, let us share our presentation.
Yes, we can see the presentation.
Okay. Good morning, everyone. My name is Munira. I am very excited to be here today. Today, I will be presenting our project on advanced in vitro techniques for potato propagation and microtuber induction. Next. Potato seeds production selective breeding and has long been a problematic sector in Uzbekistan. Below is a systematic overview of the main challenging and possible solution. Problems in the seed production system and problems related to the potato disease. This slide, we can see ongoing potato work in vitro condition. Place of work in vitro laboratory under the supervision of Professor Yun, and the main areas, subculture, apical meristem, microtuber cultivation, and we have to do recycling for microtuber production, and regeneration planting, and testing the genetic stability of microtuber. Introduction to in vitro potato culture. Purpose to cultivated potato tissues in a sterile controlled environment, and benefits disease-free plantless rapid propagation, year-round availability. Next slide, we can see apical potato propagation for flow in laboratory condition. Apical meristem isolation, obtaining virus-free initial material. Subculture, rapid propagation under sterile condition, and split process. Firstly, acclimatization hydroponics, 80 days of adapting plantlets to external environment. Secondly, the rest are placed in dark condition for microtuber induction over 4-6 weeks. About it, part of the plantlets are transferred to greenhouse, aeroponics, or to enhance root feeding and growth. Next slide, we can see methodology used in vitro potato propagation. Stage, apical meristem culture, and methodology name, virus elimination using apical meristem, key reference Wang-Hu. Subculture, nodal culture on MS medium with hormones, Murashige Cook, 1962. Microtuber induction, in vitro tuberization under dark condition, and acclimatization, gradual hardening in hydroponic system. This slide, we can see production of in vitro plantlets. Firstly, we have take apical meristem, and we have meristem cultured secondly stage, and thirdly, we can say process this during the process, and firstly, and 5-6 pictures is potato of seedlings, yeah. And next slide, acclimatization and multiplication. This one, in vitro plantlets, transplanting into greenhouse, low survival rate, and acclimatization, and the culture room. This slide is production of microtuber by in vitro tuberization. Process is picture. Thank you for attention.
Does anyone have any questions?
Yes, I have. Could you please explain this dark period for tuberization? Is it a short day or it's completely in the dark?
We have put we have put in dark room. Following is we subcultured some plantlets and we have put in dark room about it 4 or 6 weeks. And
In what temperature?
Temperature.
80 and 22 degrees.
Very, very interesting. Thank you.
This one we have choose this varieties, Chubek, Sante, and other varieties. This varieties, Chubek Sante, is the best varieties for our US laboratory.
And how do you grow these mini tubers forward?
Microtuber.
Microtubers, yeah.
And they were studying it.
This you can see this process. We have put subcultured this one, and some seedlings we have put subculturing system, and other side, yani upper part subculturing system we have put, and the down part we have put dark room.
So in the dark room is short day, 8 hours. It's not completely dark. It's only
100% karanlığı mı yok nedir?
100% ne dedi? 100%.
It's totally dark room. It's totally dark. In the, any kind of light.
Thank you very much.
Good, yes,
Thank
thank
you.
you for answering. Any more questions, maybe?
Thank you.
If not, then thank you very much. Very interesting. Yeah, so the next one will be Paula Orus and Corina Catan from Romania. So we have more guests. So Paula and Corina, are you here? Hello, Paula or Corina from Romania, are you here?
She was all morning here.
Yes. She doesn't look. Then I don't know. She's not here.
Maybe we can switch from the.
Yes, well, these presentations were really quick.
Yes,
So you want to switch on the next one or no?
Yeah, I think.
Okay, maybe then while we are waiting our colleagues from Romania, maybe Francisco can present or Maxim, if any of you are there. Yes, maybe the colleagues from Spain can present if you are ready.
Hello, I'm not ready yet, but maybe in 10 minutes. Because I'm having problem with my Wi-Fi.
Okay, then.
We connect and we'll find out what's happened with the power.
Well, theoretically, she needs to present only after 25 minutes because we had really short presentation.
What's the source with your colleague who will present about this smart agriculture? Maybe we can, while Xavier from Spain looking for Wi-Fi, we can present. Jā, Kristaps jau nav atrodas, tēvam mums vai kādā sūda.
Viņus mežiem aizbrauc.
Kurš stāstīs?
Viņi atbrauc mums.
Wonderful. What the Robert? Not that.
Me?
Nice.
I mean, okay. Then the next speaker will be me. This is it's very surprising, but okay. Okay. Then we can start. Yes, hello everyone. My name is Robert, and I will be talking about the impact of elevated CO2 content on microgreen biomass, anthocyanin and phenol content. So microgreens are small vegetable or herb plantlets that are filled with nutrients, with various vitamins and minerals and also bioactive compounds. And they have a higher concentration than mature leaves or the whole plant. And they are generally used in culinary and as a food supplement. So the purpose of this work was to increase the microgreen biomass, phenol and anthocyanin content by increasing the CO2 concentration in the atmosphere. Because CO2 is used in the photosynthesis equation, and plants use CO2 for generating carbohydrates, which they use for energy in development. So the main objective was to grow microgreens in a normal and elevated CO2 conditions. Then the fresh and dry mass was determined, and also determined the anthocyanin and total phenol content. And the first hypothesis was that the elevated CO2 should increase the biomass and anthocyanin phenol contents. So this is our experimental design. So there were two climate chambers, one with 400 ppm CO2, which is normal, and one was elevated, 1200 ppm. And after growing, the microgreens were harvested and fresh and dry mass was determined. Then a part of the fresh mass was lyophilized and used in extract preparation, and the extracts were used to determine total phenol contents using the Polnick-Chikotov method and anthocyanin determination using pH differential method. So the materials and methods, we used these climate chambers to grow the microgreens in. One had normal, one had elevated CO2 atmosphere, and the environmental parameters were the same for both chambers. The temperature was 22 degrees, relative humidity 60, and the irrigation was done once a day using tap water, so not any additional fertilizers. So the growing process we used 8 different species from Brassicaceae family as microgreens, and they were grown inside of the plastic trays using hemp mats. So each tray had 6.4 grams of seed. The seeds were germinated in total darkness for 4 days. On the 5th day, the light was introduced, and they were growing for 5 days. So the time from germination to harvest was 9 days. So they were harvested with scissors. The fresh mass was determined. Then 10 grams of the fresh mass was put in the freezer in minus 20 degrees until lyophilization. Rest of it got dried in 60 degrees for 3 days, and after that the dry mass was turned. Extract preparation. So yes, the fresh mass was frozen and lyophilized. The material was ground into fine particles. And then 10 milligrams of the dry material was put in test tubes together with 5 milliliters of ethanol. Then the mixture was put in ultrasound to extract the active compounds. Then it was centrifuged, and lastly it was filtered to get a clear supernatant. And so this extract was used to determine phenols using Follinger-Coulter method. We used 96 well microplate. So basically all of the wells were filled with water and extract or gallic acid. Gallic acid was used as a reference. And then the Follinger-Coulter reagent was also added. It incubated for 1 hour to get the reaction, and this whole plate was put into a spectrophotometer, and it really fastly got the absorptions of each well at 765 nanometers. And the absorption values were used in an equation to determine the total phenol content. The anthocyanin determination was much easier. Just had 2 cuvettes with 1 pH and the other one 4.5 pH. Then the extract was added, mixed, incubated for 20 minutes, and the spectral values of 520 and 700 nanometers were determined, and the values was used in the calculations. So for data analysis, we used computer program R with ANOVA and Kruskal-Wallis test. Then the data was also tested for normality with Shapiro and Lagoon test. Also, Tukey AHD test was used to compare the groups. So here are the results. On the x-axis, you can see the 8 different species. On the y-axis is dry and fresh mass. Blue is normal CO2, and red is elevated. So basically, the elevated CO2 concentration had no effect on neither the fresh mass or dry mass. There are differences between species on the mass because each tray had 6.4 grams of seed, but the seeds were different sizes. So some trays, depend on species, had more plants than others. So that explains the variety in masses. So the total phenol content, again, the CO2 has no effect. However, this data is useful because it shows the total phenol content of these different microgreens. And here you can see the red cabbage has the highest content of phenols. And also kale, green and red mizuna also had relatively high concentrations. The same with anthocyanins. There are some differences, but the statistical analysis conducted that there's no statistically important difference between the elevated and normal. And also, the red cabbage and red mizuna show the greatest values of anthocyanins. However, the kale and green mizuna show the lowest. So these results were very interesting because it did not match with the literature online. because here are like three researchers that conducted experiments also with microgreens with elevated CO2, and all of those experiments showed that CO2 increased at the biomass. So the question was like, why our experiment didn't work? So upon analyzing these publications further, we realized that there are three main differences. One is fertilizer, second is growing time, and third could be light intensity. So about fertilizer, in previously mentioned research, they used fertilizer to grow microgreens. We used only tap water. So the hypothesis was that microgreens require additional nutrients to assimilate the elevated CO2 from the atmosphere. So a small experiment was conducted. Red mizuna was grown in a low-high concentration fertilizer with plain water as control. The red mizuna was picked because it had the most phenols and anthocyanins, and yeah, the tree replicates. So the results indicated that still elevated CO2 concentration had no effect on biomass. However, there were statistical differences between the control and high fertilizer mizuna, which means that, yes, in order to maximize the microgreen yield, it is nice to use some fertilizer. Then the second was the growing time, because experiments in the literature were harvested on day 14 or right when the first true leaves emerged. And we harvested on the 9th, just before the first true leaves emerged. So the hypothesis was again that microgreens begin to assimilate maybe when the first leaves emerge. So again, the red mizuna was grown and harvested on days 11, 14, and 21. And yes, additionally, anthocyanin content was determined. So here are the results that, yet again, these elevated CO2 had no effect. However, this data is interesting because you can see how the anthocyanin content drops. And from this data, you can conclude that for anthocyanins, you can harvest the mizuna around day 14 to maximize the anthocyanin yield. So the third and lastly, the experiments used higher lighting intensity than we did. And since the photosynthesis equation is like carbon dioxide plus water plus light energy, that gives the reaction in chlorophyll for sugars, oxygen, and water. So this last hypothesis is that microgreens require higher light intensity to get the CO2. And this experiment is to be continued. It's for the future. So yeah. So in conclusion, the red cabbage and red mizuna has the highest phenol and anthocyanin contents, making them the best for bioactive compounds. Then fertilizer significantly increases biomass. You can use it to gain higher yields of microgreens. And to solve the mystery why the CO2 has no effect in our experiments is that, yeah, we have to conclude additional experiments with higher light intensity. So, yes, I would like to thank the project, the production of healthy plant material, for financing this. Also, thanks to Bulduri Technical School for giving me the opportunity to make experiments with microgreens. Then special thanks to Ravindu Peta from Latvian Biotechnology and Science University for helping me do the continuing to do the experiment with red mizuna. And this is the literature I was talking about in the discussion. So yes, thank you for your attention. Any questions? Oh, please. Yes, no? Okay, thank you very much. Okay, so how's it going for our colleagues in the Zoom meeting? Is Paul or Corina from Romania here? Or
Karina, Paul will be in the minutes. Some student met here accidentally. But Karina write me email if she felt ill, so she can't participate. And some problems are in Ukraine with Maxim as well.
Okay.
So two participants fell out.
Yes.
Hello,
But-
everyone. I am here.
Okay, great. I will stop sharing and yes, you can share your screen with us.
A minute, please. It is okay. Could you see my presentation?
Yes, we see it. Yes, everything seems to work.
Okay. Well, good afternoon. My name is Paula Oros, and together with my colleague Corina Catana, I am honored to present our recent research entitled Recent Developments in Tissue Culture Technique for Commercial Propagation of Heuchera species. This work was conducted in the Department of Micropropagation, Faculty of Horticulture and Business in Rural Development in Cluj-Napoca, Romania. So let me begin with a quick overview of the Heuchera genus. Commonly known as coral bells, these perennial plants are highly valued in gardens for their stunning foliage and colorful blooms. They belong to the Saxifragaceae family and include over 45 species. Their ability to a wide range of climate zones, specifically zone 4 to 9, makes them a favorite for ornamental landscaping, especially in temperate regions. Traditionally, Heuchera can be propagated by seeds or clump division. However, these methods are not always efficient for mass production or maintaining uniformity. What this is our focus on micropropagation, a method that allows large-scale, rapid, and reproducible production of genetically consistent plants. The goal of our research was to develop an efficient protocol for in vitro propagation. To achieve this, we worked with two ornamental genotypes as model, Heuchera viosa silver gumdrop and Heuchera hybrida timeless orange. The research was performed in our laboratory. We use for biological material healthy plants approximately 6 months old as the source of biological material. Specifically, we harvested stems, buds for use as explants. The explants were disinfected using 20% sodium hypochlorite solution for 20 minutes. And the clean explants were placed into the culture medium. In the initiation, we used two types of culture media, Murashige and Skog, and Murashige and Skog with Gambot vitamins. And we also compare solid culture system with the medium is where we can use gel right or to liquid system where we use Lufa cylindrica support, a sponge-like support. For rooting, the rooting was induced in vitro and ex vitro also. Using in vitro, we use MS media supplemented with 1 milligram of acid and for ex vitro routing was performed concomitant with acclimatization in jiffy pellets and in a mixture of peat and perlite. Regarding the culture initiation stage, was showed that regeneration rates varied depending on the genotype and culture medium. For example, MS medium without growth regulators was achieved the best regeneration across genotypes. In Heuchera tissue culture, the use of MS culture medium supplemented with Gamborg vitamins enhanced in vitro proliferation primarily due to the optimized nutrient profile and vitamin adjustment. This combination has been shown to improve proliferation rates and quality of shoots formation in various plant species by promoting robust tissue development and reducing stress on culture cells. The concentration of 2 milligrams per liter benzene aminopurin strongly affects the proliferation of Heuchera plantlets, leading to vitrification. So vitrification or hyperhidricity is a common problem in tissue culture when a high concentration of plant growth regulator, in our case 2 milligrams per liter benzylaminopurin, are used in the culture medium. In this case, benzylaminopurin promote rapid cells proliferation but can disrupt cellular water regulation, leading to excessive water absorption by cells, the weak cell walls, and the transpiration is altered. Regarding the proliferation, while liquid system result in higher rhizogenesis, they did not improve shoot proliferation rates. The analysis of the results show the preference of each genotype for the composition of the growing medium. For example, Heuchera viosa silver gumdrop for MS with Gamborg vitamins and Heuchera hybrida timeless orange for Murashigan school. In order to improve the efficiency of multiplication protocol in Heuchera and to reduce the cost related to micropropagation techniques, a comparative study was carried out to induce rhizogenesis in vitro or ex vitro in the same time with acclimatization. Especially since the plantlets have shown rhizogenesis capacity since the multiplication stage. The acclimatization process took place simultaneously with ex vitro rooting in acclimatization chambers. Our rooting and acclimatization process showed over 80% success rates using jiffy pellets and in the mixture of peat and perlite. This is a strong indicator of the protocol's practical viability for commercial propagation. Analyzing the interaction between the two genotypes of Heuchera and the rooting substrates ex vitro respectively, acclimatization, superior survival rates under ex vitro conditions were observed for plants growing in jiffy pellets compared to plantlets grown in the mixture substrate. The genotype Heuchera hybrida timeless orange showed a survival rate of 96% in jiffy pellets, higher than the other experimental variants. In conclusion, our research demonstrates a reliable and efficient in vitro propagation method for Heuchera species. This protocol can significantly aid in the commercial production of ornamental varieties, ensuring uniformity, health, and faster market readiness. Moreover, it offers a practical tool for the introducing new genotypes into the ornamental plant industry. Thank you for your attention, and now you are welcome to address any questions if you have.
Thank you. Any questions?
Yes, I have a question. I'm very excited that we have ornamental plants because gardening and horticulture without ornamental plants and only with potatoes, as mostly of us present, isn't the gardening. I'm very interested, how do you find out to use loofah support and how it's work, and are there no some issues about extra possibility and fungus in the growing media, and sort of a little bit explain about this.
Yes, it's a very good question. We don't have any problems with contamination because we use dry loofah. That kind of loofah, which is a substituent for a sponge. Do you know that kind of loofah? And we only cut them in small issues like 1 centimeter to only to put it in the test tube. And then we also put the liquid culture media, and we co-autoclave with this support the liquid media in the same time with loofah in the test tube, and then we use it.
And loofah was better but without
Sorry?
loofah. And loofah was better as without.
We use this kind of system in Heuchera and in Stevia, for example. In Heuchera, it was efficient for rooting. But for example, in Stevia, it was efficient for shoot proliferation. It depends maybe by the species or the combination of plant growth regulators. But if you want to have a substitute for gelling culture media, you can use this kind if the species is if the species is suitable.
Yeah, very, very interesting. We think maybe we can try some. We have now a lot of loofah in our shops and gardens, unfortunately. But it will be interesting to check because we use these plastic materials to take support for aeroponic potatoes, as example. But maybe we can try with organic ones as loofah.
Yes, sure.
Yeah. Okay. Thank you very much. Very, very
Thank you
nice
too.
presentation.
Thank you.
Yes, thank you. Okay, if that is it, then we have 15 minutes left. Theoretically, we can do another one, but I think it's better to leave it later because we.
If Francisco is ready, so we can forward with him.
Okay, maybe Francisco is ready, or
Yes, I'm
you want to
ready.
present
You
it
hear
after?
me?
Yeah. Yes. We see you.
Can you hear me?
Yes. Everything's good.
Okay, I will share my screen.
Yes, we see your screen. Yeah.
Okay, so I can start now. My name is Javier Alfosea Simon. I'm from the Spanish Research Council, where I'm doing my PhD. And I wanted to share here a part of my research plan, which title is Electrotherapy as a Potential Tool for Pathogen Elimination in Apricot Plants. So my research is focused on apiculture because it's a very important species in our country and also very important in our region, in Murcia, which most of the production is here. So, but this crop and other prunus species are facing now several important pathogen infections, such as the plant pop virus or the handstop viroid. This species, this pathogen, as you can see in the picture, can harm the fruits, decreasing the quality, and also making them not commercial. And also they can affect the yield of the farms. So it compromises the economical viability of prunus or apricot producers in Spain. One problem of both pathogens is that in some cases, agricultors are using in the beginning plant material that is not free of this pathogen. So the focus of my thesis is to establish an efficient protocol for elimination of pathogen in vitro, so it can be provided a healthy plant for the agricultors. As you know, in vitro culture can help you with that. Using meristem rescue is a common technique for plant sanitation in vitro culture, but in the case of this both pathogen, this is not enough. So we can add like additional treatment to increase the fish to find against this virus and viral, such as thermotherapy with high temperature, cryotherapy with low temperatures, chemotherapy, or in this case, which I'm going to share you, is electrotherapy. Electrotherapy is not just explored in prunus species, and also the mechanism behind this technique is not really yet understood. It is believed that the high temperatures can affect the virus, or also the electric treatment, the electric current, can harm the protein of the virus. Even though it's not yet understood, there are many articles which have used electrotherapy as an efficient treatment to eliminate both virus and viroids in many other plant species. So I wanted to explore this technique for my PhD. But we weren't very familiar with this technique, so from previous conference, we knew that Liva from this center had experience with electrotherapy, and she was able to implement it successfully in another plant species. So thanks to the copy-3 course action, I was able to work in this center for a few weeks to learn this technique, adapt it to apricot, and adapt it to apricot. So once in Latvia, I was able to perform the technique, which is very simple. As you can see in this slide, we need to use infected material. Then we place it in an electrophoresis tank which contains a liquid solution of a salt with salt. And then we have to perform surface disinfection to be able to do the meristem rescue in a sterile condition. So after that, we can evaluate if the plant is free of the pathogen or not. For my first approach to this technique, we weren't very sure how to perform this, because we can also alter the time that the plants are submitted to electrotherapy, but also the electric treatment. So we wanted to try at first the treatment that Liva knew that were effective for her plant and pathogen, and try it with apricot. For me, I'm using two varieties of apricot. One is called Canino, another is Mirlo Rojo. And Canino is infected with the virus PPV, and Mirlo Rojo is infected with the virus. So we tried 3 different electric treatments for 10 minutes each, which were 10, 20, and 30 milliamperes. Here you can see some picture of the process. This is the electrophoresis tank with the liquid solution and the shoot floating there. Then the box is closed and electric treatment begins. Then we perform the surface disinfection to perform in a laminar hood the meristem rescue. But after the meristem rescue, which is a technique when you isolate the apical part of the shoot, for this plant, I usually do it for 1 millimeter or 1.5. And then you need to grow them to get a new shoot back, which is a very long process. Once there's first we start with the initial shoot, and the second picture you can see meristem rescue 2 weeks old, and after 8 weeks I get a new shoot back, and also I have to wait after that like I have to wait since the beginning 60 weeks to perform the RT-PCR to be sure that the pathogen is eliminated without the risk of getting false negatives. For my preliminary results, here you can see the survival ratio of the meristem and also the elimination ratio. And for first variety canine infected with the PPV, we can see that the survival was good for almost all the treatment, with 70 for the first with 10 milliampere, 50% for the second with 20, and 80% for the 30 milliampere treatment. With elimination was moderate, with 14% in the first treatment and 22% in the second. For the viroid, we weren't able to eliminate this pathogen, but also the survival rate was quite good. So that suggests that we can work with more intense treatment, which is what I'm doing now. And since back in Spain, I'm working with, in this case, with the same variety infected with the virus and the viroid, and I'm using longer and more intense treatments, like 30 or 60 minutes, or even 120, depending if the survival is good, 50, 100, or even 150 milliamperes. And also, since my main goal with this technique is to mix it with other techniques, I wanted to try if I could perform this whole process into a sterile condition. For that, instead of disinfecting the chute, we wanted to try to disinfect the electrophoresis tank. For this purpose, we add inside the tank, the electrophoresis tank, for 30 minutes a solution with sodium hypochlorite, and then rinse it with water. After that, we let the electrophoresis tank in UV light for 30 minutes. I have tried this and I don't get in any contamination, so we are taking this approach for future experiments. And also I was able to perform the 60 minutes for 100 and 150 milliamperes, and the survival rate is going good, but I'm not yet finished in the process of getting a new chute back. So my main goal with my thesis is to use nanotechnology for plant sanitation and for plant validation. And using silver nanoparticle, which is a new tool that is gaining more importance in vitro culture. It is known that it can be used for disinfection of the plant material before introduction into in vitro condition. It has proven to be effective to enhance micropropagation in many species, and also can be used for against microbial infection for fungus, bacteria, and also for virus, which is one of our more recent discoveries. So with the use of silver nanoparticle, I was able to establish an efficient protocol for eliminating the virus PPV using a temporary measuring system with liquid media. But it has not been effective against the viroid. So we believe that maybe combining silver nanoparticles and electrotherapy could be a good approach to eliminate this pathogen, which is why I'm currently working now. So for a short conclusion, electrotherapy has proven to be an easy technique to apply and low cost, which make accessible option for eliminating on pathogens. And my preliminary test is happening to be moderately effective for the PPV, but it has not shown satisfactory result for the viral. However, the high survival rate that I was able to get suggests the possibility of more intensive treatment, which could improve the efficiency. The approach of doing the application of sterile condition during the whole treatment makes it a good complement for other techniques, such as silver nanoparticles or aging pills. And I'm currently working in determining the best conditions for eliminating the pathogen without compromising the survival of the meristem. So I want to thank my research group in Spain and also the center in Latvia for hosting me there for 2 weeks. And if you have some questions.
Any questions? Okay. If there aren't any questions, then yes, thank you very much. You were the last speaker of this break. So now we will have a break for half an hour from 1 to half 2, and then there will be master class of
I
the
think
case of Paul.
we go to the kitchen and
Yes.
let's see what's happening. with
Yes,
Paul.
and for the online people, I guess you will have a big break of 1 hour. And we will meet again at 14 o'clock. Yeah.
Okay, thank you.
Okay.
Thank you.
Does the floor?